Okay. Hello, everyone. Thank you so much for coming to my presentation today. My name is Shraddha. I'm a third-year CMDB major. I'm one of the undergraduate researchers in Dr. Sladig's lab. So today I'm presenting my emerging research that I hope to continue during my time here at UCR, titled Identification of Alternatively Spliced Genes in Metabolic Disease Pathways. So just to start, I'd like to talk about just the increasing incidence of obesity and other metabolic associated diseases. So according to the CDC, as of 2012, more than 1/3 of all US adults do suffer from a metabolic syndrome. This includes obesity, diabetes, and much more. There is a predicted further increase in the metabolic syndrome as the age of the nation increase, the older population increases. And so this really makes it a very critical problem to address and to improve on. So the existing treatments that have saved countless lives have definitely just addressed the symptoms of these diseases, but they haven't really gone into the metabolic dysregulation that occurs when these diseases take place in the body. And so that's what my project aims to do. I want to understand the dysregulation that occurs by studying alternative splicing. That's what I'm going to go into later. Just to begin, to first understand metabolic disease, you really have to just understand the most important metabolic organ in our body, which is the liver. The liver is very critical to maintaining homeostasis of metabolism in our body, and all the signaling pathways are very tightly controlled for this reason. During prolonged fasting periods, we see glycogen breakdown, ketogenesis, and during a fed state, we often see fatty acid synthesis and a repletion of these energy stores. So it's very helpful to understand these two states, primarily because my project studies alternative splicing in mice that are going through these two states. And it's also much easier to see the dysregulation that occurs during disease if we can look at these states. So on the bottom right corner, we see that Hnf4 alpha is highlighted in the diagram. That's because I'm going to be looking at this specific transcription factor and regulating the metabolic disease processes. So to provide a little more detail about how I'm going to investigate my project, I want to start by introducing Hnf4 alpha. It's a major regulator in the liver. It's a transcription factor that basically is responsible for a lot of the metabolic processes that the liver itself carries out. And we've noticed that when Hnf4 alpha is dysregulated, we see a lot of the, glucose homeostasis mechanisms messed up, and a lot of hepatic defects. And so it's really important to consider the role that Hnf4 alpha has in these diseases. And the two mice models that I'm using to study this are a wild type mouse that expresses the wild type version of Hnf4 alpha on the left, and an alpha 7 isoform of Hnf4 alpha on the right. The only difference between these two mice is just the kind of promoter that they express. So on the left and the right, we can see that they're shown by two different colored promoters. The left is shown in the blue promoter and the right is shown in the red promoter. And so one question you may have is just, why do we even need to look at this other isoform that's expressed of Hnf4 alpha that's expressed in this mice? And that's mainly because in the normal adult liver, we don't see the alpha 7 isoform. We only see it in high fat diets. We see it upregulated in liver cancer. And so while we don't know the exact role of it, it really seems like a relevant model to study metabolic diseases in. And I'm going to move on to discussing, why I keep bringing up alternative splicing. What's its importance? So alternative splicing is a process that's essentially the reason why we have so much protein diversity in our body. We can start out with 25,000 genes, but end up with 90,000 plus proteins. And so the way I like to think about it is as Legos. So in the picture on the right, we do see pre-mRNA, which is like a gene product, and we see that it's been spliced, so arranged in different variations like Legos can be to promote, to create different products. And these products are just proteins. So here, alternative splicing is very regulated in our body just because, again, you're producing proteins, and proteins are essentially what carry out all the cellular functions in our body. So if a metabolic disease were to take over or alter the splicing, they could produce protein products that would essentially help the disease progress. And so that's why that's why alternative splicing is just so important to study. Almost 95% of our multi-exon genes also undergo alternative splicing. So if we can identify alternative splicing events, we may be able to clarify disease pathways. And this slide, I'm basically trying to explain these questions. So first, does the fed fasted state impact splicing? Does the Hnf4 alpha isoform impact splicing? And which alternatively spliced genes are implicated in the metabolic disease pathway? Here is my workflow. So the way that I plan to identify alternatively spliced genes is first using transcriptomics data from male mice that we already have, our lab already has, and a bioinformatics program known as DEXseq that has provided us with a list of 177 genes that it believes are alternatively spliced. And I'm going to have to go in and manually curate this to double check that the program didn't call false alternative splicing, and then design primers first in silico, and then test them in the lab, and hopefully at the end of that project, be able to confirm one or more alternatively spliced transcripts predicted by that program. So I'm going to go through a bit more detail about how I'm going to do this. Here on this slide, we see a DEXseq map. This DEXseq map is the statistical tool that I mentioned using before. It essentially just aligns RNA reads, RNA sequences, and normalizes this data, so it's much easier to see. So here we see the blue line which represents the wild type mouse, and the red line, which represents the alpha-7 isoform mice. And so what the program has been that it sees a specific gene. So this gene that I'm looking at is called NR1I3. And it's seeing that there's huge differences with the way that the same exon here is being used by these two different mice models. And so it's calling them as alternatively spliced. And so I have this initial belief to go off of, but I really have to compare it to the actual gene to double check that it didn't falsely call it as alternatively spliced just because it's seeing huge differences or huge expression levels. I'm going to do this by going into a program known as UCSC Genome Browser. I can see the entire NR1I3 gene here, and what I'm going to look for is to double check that those huge differences are really what I see on this gene. One of the things I want to point out is why manual curation is so important is just because on the right-hand corner in the box, you can see that there's another gene, the TOM gene, that's overlapping with this NR1I3 gene. So this program may have actually been calling is alternatively spliced when in fact it was just two gene regions that were overlapping, and now it's just seeing this huge level expression, but it's not really alternatively spliced. And so this process allows me to mark off this gene as something that I should not consider, which has been happening a lot right now, going through that huge list of 177 genes. And so I developed a criteria going through multiple genes of what to look at and to identify which genes are alternatively spliced. So I'm going to go really quickly, just go through how I designed these primers in silico. So this is an example gene, so just Hnf4 alpha. I'm going to identify a specific region like the exon of interest and get the DNA for that exon. I'm going to be able to then use this sequence to design primers and test my primers. And if my primers do match, I'll get a successful result, which will be seen as this. And if I know it's successful, I'm able to test it in the lab. And in the lab, I will carry out RT-PCR and then test these samples on the gel. And if I see a band size that matches the length of the gene length of the exon that I'm interested in, then I know that my experiment was successful. But if I see too short of a band, I'll know that my primers did not work. And so I'd have to redesign them. So by the end of my project, I really hope to, first of all, design primers for alternatively spliced genes, which can hopefully be used to further understand metabolic disease pathways and help experiments in those pathways, as well as hopefully develop more targeted therapies for metabolic diseases, which is definitely further down the line. But again, this is just contributing to that work. I definitely want to thank my PI who spent hours every week going over my project with me and other projects that I've done in the past, Dr. Francis Sladek, and Jose Martinez, who's a great grad student in the lab who's been providing all this data and working with me to understand it, as well as other members of the Sladek lab and the UGR symposium hosts. Citations. Thank you so much.
Thank you very much, Shraddha. Congratulations. That was excellent.